And so continuing on, we're looking at what we call the neuromuscular junction, or we abbreviate it NMJ. It's also called the synapse. So the neuromuscular junction is the end of the axon near the surface of the muscle fiber or muscle cell at its motor end plate region, and it remains separated by a synaptic cleft or gap. In other words, right here, it does not actually touch the muscle. So this is a synaptic end bulb of the axon terminal, okay? And again, it's not actually touching the muscle cell or muscle fiber itself. There's a gap in there. Now, what we're looking at here, and this is electron micrograph, we have a motor neuron. These are all the branchings of the motor neuron. Okay, and you can see that it's going to individual muscle cells. We can also see a capillary and a Schwann cell. I won't get into too much detail about that, but it does wrap kind of an insulation or protective layer around the neuron. We'll talk about that more in the nervous system lectures. Okay, so let's look at the structures of the neuromuscular junction region. We have the synaptic end bulbs, which are swellings of the axon terminals. And you can see those swellings. And then the end bulbs are filled with synaptic vesicles, which are filled with acetylcholine. And the motor end plate membrane contains about 30 million acetylcholine receptors. And so again, we can see the synaptic end bulb. We see the synaptic vesicles containing the acetylcholine. The gap here is the synaptic cleft. And then we have the motor end plate. And again, this is where we're going to find that 30 million acetylcholine receptors. So as the acetylcholine is released, then it's going to bind to those ligand-gated sodium ion channels that when they open, sodium rushes in, we get an action potential running across the muscle sarcolemma. So let's look at the pharmacology of the neuromuscular junction. Now we have botulinum toxin, and this blocks release of the neurotransmitter at the neuromuscular junction, so the muscle contraction cannot occur. And we're going to find that coming from a bacteria. And typically, one place that you find it is in improperly canned foods. And actually, the bacteria is called Clostridium botulinum. Clostridium botulinum. So to give you an idea of some of the clostridiums out there and what they cause, you have, again, Clostridium botulinum, which causes botulism, which paralyzes the muscles, and you get what's called flaccid paralysis. You have Clostridium tetani. Guess what that causes? Tetanus. Does the opposite, where all the muscles contract but can't relax. You have Clostridium perfringens, which causes gas gangrene. And many of you have probably heard of C. diff. Well, C. diff is short for Clostridium difficile, which causes, a horrific colitis and creates what's called pseudomembranous colitis is what it causes, and you're passing this bloody membrane, pretty nasty. Okay, so that just gives you an idea of what the clostridiums do. They are anaerobic, which means they grow where there's not a lot of oxygen. Okay, actually, where there's no oxygen. They do not like oxygen at all. When there is oxygen present, they form spores and become kind of dormant. But when the environment's right, when the oxygen is not there, that's when they flourish. And the thing about botulism, or that Clostridium botulinum, again, creating those spores, we can accidentally introduce those spores into food, but typically the food is sterilized through the canning process. If that's not done properly, then the oxygen is purged basically from the food. And so we have an anaerobic environment in there, which is perfect for the botulism organism, the Clostridium botulinum, to flourish. Now here's something. If you take garlic, for instance, and I don't know if anybody listening does this, but chop up the garlic, put it in some olive oil to infuse, some oil, some of that garlic flavor into the oil. Here's a problem. Where do we find Clostridium? Typically in soil. Well, that garlic, since it's a bulb and grows underground, can sometimes get these botulinum spores, and you've introduced it into oil, and it's sitting there at the bottom of olive oil. How much oxygen do you think there is in the bottom of olive oil? Well, the answer is probably next to none. Actually, I would say none. And so again, you've created the perfect environment for that organism to start growing. And again, you can get botulism from doing that. Now, that's not to say you can't chop up some garlic, fresh garlic, use it in some olive oil and then use it right away. You could do that. It's when it sits there for a while and allows that, and I'm talking, maybe, a few days or what have you, but it'll allow that Clostridium spores to become active, and it can start creating that toxin. Okay, another thing is honey. You do not give honey to an infant because a lot of times there's botulism spores in honey. Now, our digestive and immune system can take care of that. An infant's digestive and immune system is not prepared for that, and they can contract botulism, which is very dangerous and deadly. So, just talked about how horrible botulinum toxin is and how it paralyzes muscles, and you basically die of suffocation. So let's take that toxin and inject it in our face. That's Botox. Okay, Botox is botulinum toxin. And how Botox works is just what we're saying here. Blocks the release of the neurotransmitter at the neuromuscular junction of those facial muscles that you don't want to move to give you wrinkles. So you're paralyzing those particular muscles. Botulism, or botulinum toxin, I should say, it can be used therapeutically as well with some really bad muscle spasms that won't release. That's a way to get them to release. Also with excessive perspiration. But again, it's blocking the neuromuscular junction. Okay. And here's the thing too, is if that Botox that's being injected in the face accidentally gets injected into a blood vessel, this is where we can see systemic paralysis. So, want to be careful. And then we have curare, which is a plant poison used for poison arrows, and it causes muscle paralysis by blocking the acetylcholine receptors, and it's used to relax muscles during surgery. Don't know if you realize this, but oftentimes when you have surgery, especially a more major surgery, you're totally paralyzed. Matter of fact, they tape your eyes closed, because your eyes would just naturally open if you're totally paralyzed. And they tape them shut so they don't dry out. But this is to keep you moving during surgery. And then we have neostigmine, which is an anticholinesterase agent. It blocks the removal of acetylcholine from receptors, so it strengthens weak muscle contractions of like myasthenia gravis. And it's also the antidote for curare after surgery is finished. Okay, so again, this helps to reverse the paralysis from the curare binding to those acetylcholine receptors. Now looking at muscle metabolism and production of ATP in the muscles. Muscles use ATP at a great rate when active. Sarcoplasmic ATP only lasts for a few seconds. Now there's 3 sources of ATP production within muscles. First one is creatine phosphate. Next one is aerobic cellular respiration. And then finally we have anaerobic cellular respiration. So looking at creatine phosphate. Now excess ATP within the resting muscle is used to form creatine phosphate. Now creatine phosphate is 3 to 6 times more plentiful than ATP within the muscle, and its quick breakdown provides energy for creation of ATP. It's going to be used for sustained maximal contraction for 15 seconds. It's used for things such as the 100-meter dash. Now, athletes have tried creatine supplementation to gain muscle mass, but here's the thing. Because of, remember, negative feedback, well, it shuts down the body's own synthesis. And there's been some long-term problems with not doing this correctly. But let's take a look at how it's formed. Okay, so we have ATP. Now, in the relaxed muscle, the ATP is going to release a phosphate, and we wind up with ADP and a phosphate. Now, what happens with that phosphate is it gets attached to creatine. So creatine is what we say is phosphorylated and becomes creatine phosphate. Okay. Well, how do we turn ADP back into ATP? Remember, ADP is adenosine diphosphate. ATP is adenosine triphosphate. Well, here we're at ADP. We have 2 phosphates. We need a phosphate. Where are we going to get that phosphate? We're going to get it from the creatine phosphate. So it releases its releases its phosphate. Now, anytime we snap off that phosphate, energy is released. And so that energy then phosphorylates the ADP and turns it into ATP. Now, when ATP is needed, again, that phosphate snaps off, energy is released, and that energy could be used for a muscle contraction or what have you. And then we're back to ADP. Okay. Now, again, if there's excess phosphate combined with creatine, then we store up the phosphate as creatine phosphate. So I hope that made sense. Looking at anaerobic cellular respiration. Now, anaerobic refers to no oxygen. And this might happen like if you are working out, for instance, or running a marathon or working the muscle to fatigue. basically the muscles go into what's called oxygen debt. Okay, so there's not enough oxygen present, but it can still metabolize for a while. So ATP produced from glucose breaking down into pyruvic acid during glycolysis. And if there's no oxygen present, the pyruvic acid gets converted to lactic acid. And this diffuses into the blood. If you've ever worked out the next morning, you're like really sore. Well, that's because those muscles have gone into anaerobic respiration and created that lactic acid. And that lactic acid is what gives you that sore feeling. Also, if you've ever done kind of a full body workout and you have that pain, and maybe you go in for, a massage, and they're working out those muscles and pushing it, all that lactic acid back into the bloodstream. Sometimes I've had patients who have felt nauseous because of that. And why would that be? Well, you're pushing that acid into the bloodstream and you're changing the pH of their blood rather rapidly. And again, that can cause nausea sometimes in a patient, or kind of a queasy or weird feeling. So now glycolysis can continue anaerobically to provide ATP for 30 to 40 seconds of maximal activity, and that can be used for something like the 200-meter race. Okay, so let's take a look at this. We have glucose. Now, where do we get the glucose into the muscle cell? Well, we can get it from the blood, but also we can use muscle glycogen. See, here's what happens when you ingest carbohydrates. A lot of the sugar from the carbohydrate is used by the cells as energy. What's left over can then be turned into glycogen and stored in the liver, or muscle glycogen and stored in the muscle. And if there's still glucose left over, it can be converted into fat. Okay, so not a good thing. All right. And so when the blood sugar kind of goes low, and again, the muscles are very active and they're going to need that glucose, well, glycogen is easily converted into glucose. And so the muscle glycogen can then be converted. Okay, so when glucose is broken in half in the process of glycolysis, energy is released, and that energy can produce 4 ATPs. Okay, but here's the thing. You make 4 ATPs, but it takes 2 ATPs worth of energy to break that sugar. So what's your profit? Your profit is 2 ATPs. It's like, for instance, if I buy something for I don't know, $2, and I sell it for $4, what's my profit? Well, my profit's $2. Okay, I got $4 for it, but it cost me $2, to get the product. Same thing here. We've made 4 ATPs, but it took 2 of those ATPs, to snap that glucose in half. So our net gain is 2 ATPs. From there, that glucose snapped in half gives us 2 pyruvic acids. And if we don't go any farther because there's no oxygen, that's when we produce the 2 lactic acids. Okay. Looking at aerobic cellular respiration, we have ATP for any activity lasting over 30 seconds. If sufficient oxygen is available, pyruvic acid enters the mitochondria to generate ATP, water, and heat. Also, fatty acids and amino acids can be used by the mitochondria. This is why things like the keto diet works, because the amino acids and the fatty acids are used as an energy source. And it's going to provide 90% of the ATP energy if activity lasts more than 10 minutes. Okay. So let's kind of talk about this a little bit. And again, this is very, very basic. When we bring in the glucose, first of all, we go through glycolysis. We talked about that. Snap the sugar in half. We wind up with how much ATP? 2 ATPs. Okay. If we go on into aerobic cellular respiration, then we move on down to the Krebs cycle or citric acid cycle. And there's a lot of shuffling around of carbon atoms. And then eventually we wind up with making 2 more ATPs. Okay. Then we move on to the electron transport chain. Okay, the ETC, electron transport chain. Now, this is where ATPs are going to be mass produced. But let's go back up to glycolysis and the Krebs cycle or citric acid cycle, same thing. During that time, we're also making these things called electron transporters. And they are going to transport electrons to the electron transport chain. And think about it this way. If we have a water tower and we pump water up into the water tower, we have what kind of energy up there? We have some potential energy, right? If that water comes down the pipe and runs through a pipe that maybe we put a turbine on, and that turbine spins, we can generate some energy. We can generate some electricity. And I need to patent that because I've been talking about that for the 20-something years I've been teaching. So it's kind of my idea. Anyway, so in the mitochondria, we pump hydrogen ions up against their concentration gradient. So that's like pumping the water up into the tank. Well, anything that where there's a lot of course, it's going to want to escape. Again, we pump water up into a tower. It wants to come down. And the only way it can do that is through, the main water line. Well, the only way that that hydrogen pumped up against its concentration gradient can leave is through an enzyme that's called ATP synthase. Okay, it's a channel that these hydrogen ions can go down. And it's almost like spinning a turbine, and we create what's called a proton motive force. That proton motive force is the energy used to take ADP and phosphorylate it, again, stick a phosphate onto it, and turn it into ATP. And so if we look at the glycolysis, making 2 ATPs, the Krebs or citric acid cycle, making 2 ATPs, and then the electron transport chain, making about 34 ATPs, we're looking at about 36 to 38 ATPs from one glucose molecule in aerobic respiration, as opposed to just 2 in anaerobic respiration. Okay, muscle fatigue. It's the inability to contract after prolonged activity. Central fatigue is the feeling of tiredness and a desire to stop lecturing. Oh, I'm sorry. Did I say that out loud? No, just kidding. Central fatigue is the feeling of tiredness and the desire to stop. It's a protective mechanism. We also get muscle fatigue with the depletion of creatine phosphate. And we also get it with a decline of calcium within the sarcoplasm. And the factors that contribute to muscle fatigue is insufficient oxygen or glycogen, buildup of lactic acid and ADP, and insufficient release of acetylcholine from motor neurons. And we see that in some of the motor neuron diseases. Okay, oxygen consumption after exercise. Muscle tissue has two sources of oxygen. It diffuses in from the blood. And remember, we talked about myoglobin? So released by myoglobin inside the muscle fibers. The aerobic system requires O2, oxygen, to produce ATP needed for prolonged activity. And this is why, as you work out or move or do things, increased breathing effort during exercise helps to bring in more of that oxygen and get rid of that excess carbon dioxide as you're working out. Now, looking at recovery oxygen uptake, elevated oxygen use after exercise, because if you're in oxygen debt, you're going to want to bring in more oxygen, give off that excess carbon dioxide. Also, during the recovery phase, lactic acid is going to be converted back to pyruvic acid. And as long as you have enough oxygen, that pyruvic acid can then, make its way onto the Krebs cycle. And also when you work out, you notice that you get very warm, very hot. So the elevated body temperature means all these reactions can take place faster. We're talking chemistry here. Again, if you've taken chemistry, if you want to speed up a reaction, what do? You apply some heat. Okay, looking at a motor unit. A motor unit equals one somatic motor neuron in all of the skeletal muscle cells or fibers it stimulates. So if we look at this motor neuron here, we see that it stimulates this muscle cell, and this muscle cell. Whereas this one stimulates this cell and this cell. Now, muscle fibers are normally scattered throughout the belly of the muscle. And one nerve cell supplies on average 150 muscle cells that all contract in unison. And the total strength of a contraction depends on how many motor units are activated and how large the motor units are. In other words, like walking, for instance, you're going to use many large motor units. Okay, you don't want to take that same strength that's in your legs walking and put that into, raising a coffee mug up to your mouth to take a drink of coffee. You would probably knock your teeth out, maybe knock yourself out. So that would not be a good thing. So if we're going to do some more delicate work, we're going to use small motor units and few of them. The more strength you need, the more motor units we incorporate, and that's what allows for that increase in strength or decrease in strength. Okay, so if that makes sense. Okay, looking at a twitch contraction. So a brief contraction of all fibers in a motor unit in response to a single action potential in its motor neuron, or electrical stimulation of the neuron or muscle fibers. That's like when I had my practice. If a patient, had some muscle issues going on, muscle spasm, muscle pain, what have you, I would use my electric stim unit and place electrodes on it, on the surface of the skin over the muscle. And when I apply that current to the muscle, it will cause that muscle to contract. And typically what I was trying to do, especially if the person was in, had a muscle spasm, is I wanted to fatigue out that muscle. So, we get it to contract long enough to where it would just fatigue out, and then you can break that spasm and relax that muscle. Okay. So we can either have a single action potential in a motor neuron, or we can use electric stim of the neuron or muscle fibers to generate this twitch. And we graph that twitch out on this thing called a myogram. And so the action potential lasts about 1 to 2 milliseconds, and the twitch contraction lasts from 20 to 200 milliseconds. And so we're going to look at this myograph, and we're going to look at force compared to time. And so we have what's called the latent period. And that takes place, or it takes about 2 milliseconds. Here, calcium is being released from the sarcoplasmic reticulum. And basically what we're doing is taking the slack out from the elastic components. It would be like, okay, if I was going to try to pull, I don't know, a cart, and I tie a rope to the cart. When I first start pulling on the rope, I'm not pulling on the cart yet, right? The rope is sagged down to the ground. I have to start pulling till I take all that slack out of the rope. Okay. Then we have the contraction period. So all the elastic, which I say slack, has been taken out. That's going to last from 10 to 100 milliseconds. And here the filaments are going to slide past each other. And so that's right here in the red. So we have the latent period. We're taking out the slack. No contraction taking place yet. Slack's been taken out. We have the contraction period. The contraction is taking place. Then we have the relaxation period, and that lasts about 10 to 100 milliseconds. And the muscle is not going to relax, like boom, where it's totally relaxed all at once. We have to pump calcium back into the sarcoplasmic reticulum. It takes active transport to do that. You're not just going to suck it all in at once. So instead, we're going to get this curve here. Okay. So this is the relaxation period. Then we have something called the refractory period. Here, the muscle cannot respond and has lost its excitability. And it's about 5 milliseconds for skeletal muscle and 300 milliseconds for cardiac muscle. So a portion of that relaxation period is where you have that refractory period. So no matter how much stimuli you give to the muscle, it's not going to contract if it's in refractory period. It's like, I don't know if you've ever seen these flash units, on like the single reflex cameras, the larger cameras. Well, when you would take a picture with that, the flash would of course flash, and we would call that depolarization if we were talking about muscles or nerves. Now, if you tried to take a second picture right away, you would not get that flash. Okay. Or if you press the button on the back of the flash unit, because there's a little test button, again, it would flash, but if you pressed it again right away, it wouldn't flash. And you can hear this little sound as the battery is recharging back up again. Okay, I think what's in there is what we call a capacitor. Capacitor pulls in, stores energy so that it can give a big release of energy at once. So it'll give out a bigger blast of energy than what the batteries actually have. Anyway, so it charges up, and so that period of time where we're pressing the button and nothing's happening, or trying to take a picture and nothing's happening, that's the refractory period. Okay. Once it's out of the refractory period, then boom, it'll flash. That's depolarization again. Oh, by the way, the opposite of depolarization is repolarization. So as the battery is, or the flash unit is recharging back up again, that's what we would call repolarization. And then let's take a look at wave summation. So if a second stimulation is applied after the refractory period, but before complete muscle relaxation, a second contraction is stronger than the first. In other words, they can sum up or add up on each other and stack up on each other. So here's our twitch reaction. Boom. Okay. Now if we hit it with a second stimulation, boom, here we're in refractory period. Once we're out of refractory though, and it hasn't totally relaxed yet, then boom, we can stack up another another impulse, a stronger impulse. Okay, so this is wave summation. Now in this, this is called unfused tetanus. Okay, and I can set this actually on my electric stim machine, okay, to do this. It's kind of annoying. Typically I set it up on the fused tetanus, but I'll explain that in a second here. Now, suppose someone's having problems with the shoulder. Maybe there's some swelling in the shoulder. That's what this is good for with the electric stim, where you use the unfused tetanus. So if I put it on the shoulder, all of a sudden you'd see their shoulder start twitching, and the shoulder is getting tighter and tighter and tighter and coming up toward the ear. And then all of a sudden it's at the ear, and then it fatigues out, and then boom, it drops. Your shoulder just drops back down. And then a few seconds later, all of a sudden you feel it start to twitch again. And again, it starts twitching and coming up to your ear as it's twitching, twitching, twitching. And then finally it's held in place, and then it fatigues out, goes into refractory period, and boom, it relaxes. Now in fused tetanus, if I set the machine to this and it's still attached to the shoulder, as soon as I turn on the machine, boom, you get this contraction. It pulls the shoulder up toward the ear and holds it there until it fatigues, and then it drops. And then as soon as it gets out of refractory period, it'll just pull right back up again. So I should do a video of that and show you, but I'm not going to. Anyway, so I hope you can visualize what I'm talking about. But again, this is wave summation. And so looking at complete and incomplete tetanus. So unfused tetanus, and again, that's where, the shoulder is kind of bouncing a bit. If you stimulate at 20 to 30 times per second, that's what I'd set the machine for, there will be only partial relaxation between stimuli, and there it is. And then the fused tetanus, if you stimulate it 80 to 100 times per second, again, that's what my machine is usually set at, a sustained contraction with no relaxation between stimuli will result. Okay. So explanation of summation and tetanus. Wave summation and both types of tetanus results from calcium remaining in the sarcoplasm. The force of the second contraction is easily added to the first because the elastic components remain partially contracted and do not delay the beginning of the next contraction. Looking at motor unit recruitment, motor units in a whole muscle fire asynchronously. Some fibers are active while others are relaxed, and it delays muscle fatigue so that the contractions can be sustained. For instance, sitting upright, okay, your back muscles, some of those fibers are relaxing while the others are doing the job. Okay, that would be like, let's suppose you have to hoist something up with a rope, something really heavy, and there's a bunch of you holding on to that rope. Well, if everybody just holds the rope, at full force, eventually everybody's going to fatigue, and then that weight's going to drop. But if you have a bunch of people holding the rope that are using full force and a few of the others are relaxing, then when it comes time where the people holding the rope start to fatigue a bit, then the others can take over and hold it while the first group relaxes. Okay, so you see what I mean? Otherwise, you'd be sitting there upright, then all of a sudden your back would give way and you would just slump forward and whack your head on the table. Although I think I have seen that with a few students over the years. So what's it going to do? It's going to produce a smooth muscle contraction and not a series of jerky movements. And so precise movements are going to require smaller contractions, so motor units must be smaller, so less muscle fibers to nerve. And then large motor units are active when large tension is needed. So think of your fingers, for instance. Put your finger and thumb together and look at what kind of a tiny little movement you can make, as opposed to the large, grosser movements of your lower extremities. So muscle tone is involuntary contraction of a small number of motor units, alternately active and inactive in constantly shifting pattern. Now, it helps to keep the muscles firm even though they're relaxed, but it does not produce movement. Now, it's going to be essential for maintaining posture, head upright, and it's important in maintaining blood pressure. And there's a certain tone of smooth muscles in the walls of the blood vessels. Now, looking at isotonic and isometric contractions. Isotonic contraction, a load is moved. Concentric contraction, a muscle shortens to produce force and movement. And eccentric contraction, a muscle lengthens while maintaining force and movement. So these two are isotonic contractions. So load is being moved. So this is a concentric contraction. This is an eccentric contraction. And then an isometric contraction, no movement occurs, as we see in this picture. So the tension is generated without the muscles shortening. And again, it's going to be used for maintaining posture and support objects in a fixed position.